Hello students, welcome to EPG Pathshala. I am Dr. Hari Prasad, Associate Professor at the Department of Biophysics, All India Institute of Medical Sciences, New Delhi. Today I am going to talk to you about the module Sodium Deodyl Sulphate Polyacrylamide Gel Electrophoresis, also called as SDS PAGE, from the paper Techniques in Molecular Biophysics, Part 1. What are the learning objectives of this particular module? We will be having an introduction. Then we'll talk about the protein fold and the role of SDS in SDS electrophoresis, SDS as a chemical entity, factors affecting protein stability and solubilization, polyacrylamide gel matrix, gel pores, structure and polymerization of the matrix, the do's and don'ts in SDS PAGE, discontinuous gel in SDS PAGE, Tris-Glycine-Chloride System of ions, techniques, that is the casting of the SDS gel, sample preparation and loading, visualization of the protein bands in SDS system, applications and molecular weight determination, advantages and disadvantages, and now we will start with the introduction. This particular technique, that is SDS PAGE, is widely used in the fields of biochemistry, forensic medicine, genetics and molecular biology. This particular lamellae system of SDS PAGE was first introduced in the 1970s. This has more than 2,27,000 citations as of today, with an annual average citations of more than 3,000. What is the fundamental basis of the resolution of proteins using this particular system? Molecules in an electric field move with a speed dependent on their charge, shape and size under the influence of electromotive force. When the charge distribution is the same, the molecules having higher molecular weight or size experience a more pronounced retardation in their migration, while smaller proteins migrate quickly through a particular matrix. A protein is a macromolecule consisting of a sequence of amino acids, each connected to the other by a bond called as a peptide bond. The protein consists of one or more polypeptide chains that are held together by a number of molecular interactions, resulting in several levels of organization. A functional protein therefore has a three-dimensional structure resulting from hydrogen bonding, hydrophobic interactions, salt bridges, van der Waals forces, and disulfide bonds. So therefore, for optimal separation during electrophoresis, what is required is that the secondary and the tertiary structure of the protein to be broken down. For this, we use sodium deodyl sulphate and a reducing agent such as beta-mercaptoethanol or dithiotritol. So SDS has an important role as a detergent in breaking down non-covalent interactions. It also coats the protein uniformly with a negative charge. Reducing agents such as beta-ME and dithiotritol cleave disulfide bonds. In addition, heating of the protein samples facilitates denaturation and unfolding process, allowing chemicals like the SDS and the reducing agents to interact with the protein. The end result is all the polypeptide chains in the sample become linearized and are uniformly charged to have a particular charge to mass ratio. Sodium deodyl sulphate is an anionic detergent having both a hydrophobic end, that is comprised of the deodyl group, and a hydrophilic end comprising of the sulphate group. It is therefore called as an amphipathic surfactant. The tertiary structure of most of the proteins that result from interactions are broken down by the SDS. The hydrophobic interactions are broken down by the hydrophobic end of the SDS by making interactions with the hydrophobic side chains of the amino acids, while the sulfate group on the SDS can disrupt hydrogen bonding in secondary protein structure by the same mechanism. Additionally, you also have the SDS, which coats the negative charge to the protein, and therefore the protein attains a net negative charge, due to which when an external field is applied to the gel in buffer, each protein molecule will flow towards the positive electrode. This allows the acrylamide matrix to separate the proteins based on size. The SDS covered negatively charged protein molecules move through the gel matrix. Smaller molecular weight proteins are able to navigate through the pores in the matrix more rapidly than those with a high molecular weight. Thus, proteins in the sample are separated by size and relative molecular weight irrespective of the charge. So, what are all the factors that affect protein stability and solubilization? Beta-mercaptoethanol or dithiotrietol disrupt the disulfide bonds that stabilize tertiary or quaternary structure of the proteins. Heating the protein sample facilitates denaturation, and the unfolding process allows chemicals such as SDS and DTT to interact with the proteins. The sodium deodyl sulfate solubilizes the protein by binding uniformly along their length to a level of 1.4 gram of SDS that binds to a gram of protein, giving it a very uniform electric pull. All the proteins in the sample are therefore linearized and are uniformly coated with a negative charge on their surface, and as a result, the charge to mass ratio of all the proteins are the same. We now try to understand about polyacrylamide gel matrix. The polyacrylamide gel matrix is used to separate proteins ranging from 5 kilodalton up to 200 kilodalton. What are the special features of the polyacrylamide gel matrix? They are chemically inert, they are electrically neutral, they are hydrophilic in nature, transparent for optical detection, and based upon the concentration of the acrylamide and the bisacrylamide used, you have a pore size that can be controlled. The size of the pores is determined by the concentration of the acrylamide and the linker bisacrylamide. We usually have a discontinuous sodium deodyl sulfate polyacrylamide gel electrophoresis by having two different gels. The top part of the gel is called the stacking gel, which is made with 4% acrylamide, and the bottom part of the is called the separating gel that ranges from 5 to 15%. The size of the pores in the polyacrylamide gel is dependent on two important parameters: the total solid content, that is the percentage T, and the ratio of the cross-linker acrylamide monomer, that is percentage C. The total solid content is given by the formula the sum of the acrylamide and bisacrylamide to the total volume in ml multiplied by 100. The percentage of the crosslinker is given by the amount of crosslinker in G divided by the total amount of acrylamide and bisacrylamide multiplied by 100. The acrylamide concentration and the range of molecular weights of the proteins that can be separated is shown in this particular table. The lower the molecular weight range, the higher is the recommended gel percent, and vice versa. The higher is the protein molecular weight range, lower will be the recommended gel percent. Electrophoresis of proteins take place on a porous matrix made up of repeating units of acrylamide, while N,N-methyl bisacrylamide acts as branching agent or the crosslinker. When a mixed solution of acrylamide and crosslinker monomers copolymerize into long chains that are covalently cross-linked, a polyacrylamide gel is formed. These gels are physically tougher than agarose gels, and the polymerization process is also more complex. The polymerization itself is a process that is initiated by a free radical catalyzed reaction. The most common source of free radicals is the quaternary amine NNNN tetramethyl ethylenediamine or TIMID as a catalyst, and ammonium persulfate as the initiator peroxide. Acrylamide polymerizes spontaneously in the absence of oxygen. So, the polymerization process involves complete removal of oxygen from the solution. Polymerization is more uniform if the mix is degassed to remove much of the dissolved oxygen by placing it under vacuum for 5 minutes or so before the polymerization. We initiate polymerization by adding freshly prepared ammonium persulfate to be mixed with timid. The amounts of each depend on the quality of the acrylamide used and should be determined in advance by trial and error. Do's and don'ts in SDS page. Acrylamide is a very potent neurotoxin, so students should take care when they are handling it. Rapid polymerization can cause inconsistent gel structure due to heat, and so adjust the concentration of the catalyst so that the polymerization occurs in a very controlled manner. The matrix is electrically neutral. The presence of fixed charged groups can cause the flow of water towards one or the other electrode, and usually this being the cathode. This particular phenomena is called electroendosmosis and markedly decreases the resolution or the separation. Generally, APS and TEMID are used at approximately equimolar concentration in the range of 1 to 10 millimolar. SDS PAGE uses a discontinuous polyacrylamide gel as a support medium for the electrophoresis. It comprises of two types of gels. One is the stacking gel, which is in the upper part, and then you have a separating gel, which is in the lower part. The stacking gel helps to concentrate the protein samples into a very sharp zone, and the resolution of these proteins then take place on the separating gel. Tris-Glycine-Chloride System of ions. The whole concept or the biophysical principles that is there in SDS page revolves around this particular concept. The chloride ions from the gel buffer serves as the leading ion as it has the highest attraction to the anode with respect to the other anions in the system. Glycine 2 is negatively charged, is the primary anion provided by the running buffer. It serves as a trailing ion due to its partial negative charge and remains behind the more highly charged chloride ions. The tris base is commonly ion present in both the gel and the running buffers. The gel and the buffer ions in the tris-glycine system form a pH gradient of 8.8 in the resolving portion of the gel. Now, let us try to understand what happens when we are running a SDS page gel. When we mix the proteins with the loading dye, we have it as a mixture as shown in figure 1. But subsequently, you see in the lane 2 that the chloride ions, being highly negatively charged, moves forward to create a ionic front. This is followed by all the proteins, and you have the glycine ions that are shown as red color trailing behind the proteins as shown in lane 3. Therefore, the proteins are therefore sandwiched between the leading chloride ions and the glycine ions behind. This particular concept introduces an added electromotive force to take them into the resolving gel. At the resolving gel, because there is a change in the pH to 8.8, the glycines move ahead of the proteins as shown in the lane 4, and because of this, the proteins will then come behind the glycine. Once in the resolving gel, as shown in lane 5, what happens is, because all the proteins are uniformly charged by SDS, the parameter of molecular weight or size then dictates the separation of the proteins in the resolving gel as shown in figure 6. Thereby, separation of proteins is now possible and the resolution is clearly demarks the proteins as separate bands. To give you a particular example, it is akin to boys running across a particular field. With the positive motive of running from one end of the ground to the other, small or little boys will be able to crisscross through all the adults and reach the other end. In much the similar way, in a matrix that is caused by the acrylamide, larger proteins tend to have a frictional drag and are retarded, whereas the smaller proteins crisscross between the sieves to reach the other end. We now discuss the steps that are involved in gel casting. We need to assemble the vertical slab gel apparatus using a 1 mm spacer, making sure that the plate is thoroughly cleaned and dried before use. We need to seal the glass plates on the side with agarose or have blockers. We need to pour the separating or the resolving gel mixture to a level of approximately 2.5 cm below the glass plate surface. Immediately after pouring the gel mix, it must be overlaid with water-saturated butanol to an additional height of 0.5 cm or so, and butanol becomes the top layer in this particular situation. Adding butanol to a single gel will drive the acrylamide mix down, raising the level in the others. So care must be taken to distribute the butanol equally among the gel surface. The purpose of butanol is to produce a smooth, completely level surface on top of the separating gel, so that the bands when formed are straight and uniform. Butanol holds very little water in solution, forming a very neat layer on the top, which is why we use it. Water would make an effective overlay but would mix the acrylamide solution, diluting it. In fact, butanol that we use is saturated with water so that it does not dry out the gel mix. Once this is done, the next stacking gel is poured on this particular separating gel. Immediately, we then insert a comb and allow the polymerization of the gel. Polymerization can be confirmed by taking some of the gel mix into a pipette and allowing it to stand. And we then check for the polymerization after 10 minutes or so. When the gel mix can no longer be expelled by squeezing the bulb, the separating gel is said to be set. It should not take more than 15 minutes for any of the gel mixes to polymerize. In SDS PAGE electrophoresis, a particular type of denaturing buffer is mixed with the protein. This particular buffer is called as the sample buffer. We obtain good denaturation by preparing a sample to a final concentration of 2 mg per ml of protein. Sample buffer comprises of a 2x concentrate consisting of 2% SDS, 20% glycerol, 20 millimolar Tris-Glycine-Chloride of pH 6.8, 2 millimolar EDTA, 160 millimolar of DTT, or a 0.1 mg per ml of bromophenol blue dye. One prefers DTT to beta-mercaptoethanol because the latter has a much stronger unpleasant odor and doesn't denature our blood fractions very well. A pinch of bromophenol is added and serves as a tracking dye and is added at a concentration of 0.05 mg per ml. SDS, DTT and heat are responsible for the actual denaturation of the sample. SDS breaks up the 2 and 3-dimensional structure of the proteins and by adding negative charge to the amino acids. Therefore, the proteins are more or less straightened out and immediately rendered functionless. Some quaternary structure may remain due to disulfide bonding, and these particular disulfide bonds are removed using reducing agents such as DTT and beta-mercaptoethanol. DTT is a strong reducing agent. Its specific role in sample denaturation is to remove the last bit of tertiary and quaternary structure by reducing the disulfide bonds. EDTA is a preservative that chelates divalent cations and reduces the activity of proteolytic enzymes that require calcium and magnesium ions as cofactor, thereby preserving the proteins in the sample. The Tris acts as a buffer, which is very important since the stacking process is discontinuous. Electrophoresis requires a specific pH. Glycerol makes a sample more dense than the sample buffer. So the sample will remain in the bottom of the well rather than float out. The dye allows the investigator to track the process of electrophoresis. To completely denature the samples, we heat them in a steaming water bath for at least 10 minutes. After a SDS PAGE experiment, we then have to stain the gel in order to visualize the protein bands. The final outcome of the experiment is therefore dependent on the quality and the sensitivity of the dye that is used. There are many dyes that could be used for the experiment. They are Comasi blue, silver stain, colloidal Comasi, and Cipro ruby. Comasi blue is an anionic dye which binds non-specifically to proteins. The structure of this dye is mainly non-polar and it is used in methanol solution acidified with acetic acid. Proteins in the gel are fixed by acetic acid and simultaneously stained. The excess dye incorporated into the gel can be removed by using a destaining solution. The proteins are detected as blue bands on a clear background. As SDS is also anionic, it may interfere with the staining process. Therefore, large volume of staining solution, at least 10 times the volume of the gel, is ideally recommended. Comasi blue usually detects proteins in the range of 50 nanogram amount. Silver staining, on the other hand, is used when more sensitivity is required for detection and is usually 50 times more sensitive than the Comasi blue. Silver nitrate is reduced in the presence of proteins to give a brownish colored spot against white background. We then have a modification of Comasi called as colloidal Comasi, which uses ammonium sulphate to precipitate the proteins. In this particular technique, we get blue bands against a transparent white background. The sensitivity of the technique is 10 nanogram of protein. And finally, we have the Supra ruby, which results in red bands against a black background and has sensitivity of 0.25 to 1 nanogram. SDS PAGE has a wide range of applications in protein chemistry and molecular biology. They are to establish the size of the different proteins in a sample having a variety of proteins as in cell culture or tissue lysate. Second, protein identification based on size that can be done using protein molecular weight markers. The purity of the sample can be established by checking if unwanted protein molecules are present in it or not. Identification of subunit characteristics of proteins can be done. That is, if the protein is multi-subunit, then denaturing conditions could break the disulfide linkages and render the two subunits separate where it can be visualized as distinct bands on the gel. In cloning and gene expression, SDS PAGE is used to establish whether the induction of gene has occurred and whether the recombinant protein is being overexpressed. Peptide mapping. In this process of peptide mapping, controlled cleavage of pure protein with small amounts of pure protease to generate peptides of characteristic reproducible sizes is done. These peptides are then separated on page to produce a fingerprint characteristic of the protein. Peptide mapping can be used to map cleavage sites in an unknown protein or for identification of an unknown protein based upon its fingerprinting identity with a previously tested sample. SDS PAGE can be used to analyze the relative amount of differentially expressed proteins in multiple samples. For this purpose, measuring the protein levels, intensities of specific bands corresponding to the proteins of interest are measured using commercially available software after a densitometric analysis. And lastly, and most important, is the quantification of the molecular weight using SDS PAGE. The relative migration or the mobility RF of a molecule in gel electrophoresis is determined by the free solution mobility along with the sieving action of the gel matrix. In SDS protein electrophoresis, the SDS buffer uniformly coats the proteins with the negative charges, equalizing the charge to mass ratio for all proteins. Ultimately, the relative migrations are determined only by the pore size of the gel whose sieving action is proportional to the molecular weight of the protein. The relative migration for a set of standard protein markers varies inversely with the log of the molecular weight, as shown in the figure. After the standard plot is made, the molecular weight of the unknown protein or the test protein in the sample can be interpolated from the graph using their respective relative migration values. The accuracy determining the molecular weight can be confidently be expressed in the range of 5 to 10%. SDS PAGE could also be used into understanding the quaternary structural formations of the protein complexes. How so? Here we use native gel alongside denaturing gel which has components of SDS and beta-mercaptoethanol. So SDS, as we understand, breaks down the non-covalent interactions. Beta-mercaptoethanol is a reducing agent that leaves disulfide bonds. Now, let us look at the gel 1. In a native gel, you have a 40 kilodalton protein, while in the denaturing gel comprising of SDS and beta-mercaptoethanol, you have two bands of 30 kilodalton and 10 kilodalton. Therefore, it can be understood that the protein is a heterodimer. Coming to the second situation, we have a 40 kilodalton protein in the native gel, and in a denaturing gel with SDS and beta-mercaptoethanol, we have a single band of 20 kilodalton. This reflects that the protein is a homodimer with the subunits measuring 20 kilodalton each. In the third situation, we have a 40 kilodalton band in the native gel, while there are two bands on the denaturing gel comprising only of SDS without beta-mercaptoethanol. This indicates that the units have been separated out by SDS that has broken the non-covalent bonds. Therefore, it's a heterodimer that was formed by the non-covalent interactions between the subunits. In the fourth situation, we have a 40 kilodalton band on the native gel, and on the denaturing gel with SDS, we have a single band. This again reflects on the same lines that it's a homodimer linked by non-covalent bonds. In the fifth situation, we have a single band on the native gel, and we have two gels on a denaturing gel that uses only beta-mercaptoethanol without the SDS. This indicates that beta-mercaptoethanol has effectively cleaved the disulfide bonds between the subunits, thereby making it a heterodimer comprising of subunits of 30 and 10 kilodalton respectively. And lastly, in the sixth situation, we have a single 40 kilodalton band on the native gel and have another single band on the denaturing gel with beta-mercaptoethanol. Again, it indicates that beta-mercaptoethanol, a reducing agent, has cleaved the disulfide bonds between the subunits, still resulting in a single band, thereby helping us to conclude that the protein in the study is a homodimer formed by two subunits of 20 kilodalton each linked by covalent bonds. Thereby, it is very clear that SDS PAGE is a very effective tool in understanding quaternary associations in proteins. We now try to weigh the advantages and disadvantages of the SDS PAGE experiment. The advantages would be manifold. They are: migration rate is linearly proportional to the molecular weight. It's a highly sensitive method depending upon the type of the stain used and separates out 2% differences in mass. Requires small amount of samples. They are stable, chemically cross-linked, and a sturdy gel could be cast for the electrophoresis. The disadvantages could be that there's a very poor band resolution due to highly alkaline operating pH, and of course handling acrylamide itself would be dangerous because it's a potent neurotoxin. Gel preparation is cumbersome, difficult, and requires certain amount of time. And lastly, sometimes the reagents could be found to be very costly. But nevertheless, the advantages are definitely outweigh the disadvantages, making this an indispensable technique in protein biochemistry and molecular biology. So students, let us now summarize what we have learnt in this particular module. SDS PAGE is used to separate proteins based on the molecular size. The matrix used is made up of polyacrylamide and bisacrylamide as cross-linking agents. This utilizes a discontinuous gel system or the lamellae setup where two gel portions, that is the stacking gel and the resolving gel, having pH 6.8 and 8.8 are respectively used. The stacking gel has a higher pore size as compared to the resolving gel, and this helps in concentrating the protein bands along with the chloride and the glycine system of electromotive force into a narrow zone. This helps in improving the resolution of the system. The running buffer is made up of glycine, which determines the migration of proteins as its movement is dependent on the pH of the environment in the gel. SDS is employed to coat the proteins, providing them with a very uniform negative charge and then removing out all the native charge that is otherwise existing on a particular protein. Beta-mercaptoethanol and DTT are used as reducing agents to cleave all the disulfide bonds that make up or stabilize the protein structure. Thank you so much.